Today we will be solving Biology Paper 1B from Pearson Edexcel International GCSE 921, November 2025. Question number 1. We are given a diagram of amoeba cell 1A I. Which structure is the nucleus? We know that nucleus is a large organ and it will be in a darker shade in the diagram. So D is the correct answer. Which structure is the cytoplasm? Cytoplasm is the jelly-like structure. It will be in a lighter shade and this is our cytoplasm. So the correct answer will be Q. Which structure contains chromosomes? Nucleus always contains genetic informations or materials like DNA and chromosomes. So the correct answer will be D. Now we are required to calculate the actual length of the amoeba, and we will use our ruler to measure the length of the amoeba from X to Y, and here is the value 43 millimeter. We have to input this value into the formula to get the actual length. Always keep in mind in which unit we have to write the actual value in, actual length in, and we must convert this millimeter into micrometer to get the correct answer. Now give 3 differences in structure between an amoeba and a virus. So here are a few differences. Amoeba has a nucleus, as we can see in the diagram. It has cytoplasm, it has chromosomes and cell membrane. Amoeba is larger, and viruses are very tiny. Virus has protein coat, capsid, envelope, or we can also add plasmids. Question number 2. A student uses this apparatus to investigate the effect of concentration of a substance on the rate of diffusion. So here the teacher places 6 pieces of damp litmus paper at regular intervals, which is 4 centimeter. The distance of the litmus paper is our independent variable because we are changing the distance. We are controlling the distance. Okay, that's why this is our independent variable, and our dependent variable will be the time taken for the litmus paper to change its color. 1 drop of ammonia is added to a cotton wool and is placed at the end of the test tube. Here 2A, state what is meant by the term diffusion. We have to learn this definition by heart. It is the movement of particles or atoms or molecules from a region of high concentration to a region of lower concentration. Now we are moving on to 2B1. Give the dependent variable in this investigation. As I have said, time taken for litmus to change color is our dependent variable. We have to be very clear when we are writing about these variables because it's better to be clear and specific to ensure the mark. Now B2. Plot a line graph to show the time taken for litmus paper at each distance to change color. Use a ruler to join your points with straight lines. Draw separate lines for 1 drop of ammonia solution and for 3 drops of ammonia solution. So I have plotted all these points using all of these points using this table from this table, and we have to give the key, or we have to label beside the line that this line is for 3 drops of solution and this line is for 1 drop of solution. We will always keep our independent variable in the x-axis and the dependent variable in the y-axis.